Hello everyone, my name is Sharel Cornelius, and the title for my poster is Developing Effective Reagents for Tyrosine Bioconjugation. I will go over a quick overview of the work that we did. We evaluated the site-specific labeling of tyrosine residues employing PTAD-based bioconjugation reagent. Our study also provides an extensive structural proteomics investigation of labeling tyrosine in samples of varying complexity, ranging from smaller peptides to bigger proteins and moving on to much complex samples like cell lysates. We also use the tandem mass spectrometry experiments to locate and characterize the labeled tyrosine residues. The tyrosine amino acid is an aromatic essential amino acid and is present in very low abundances in the proteins. The unique structural and electronic features of tyrosine residue makes it an interesting target for covalent inhibitors under certain physiological conditions. Surprisingly enough, tyrosine has also been found in the production of neurotransmitters and has a key role to play in various important diseases. For example, the SARS-CoV-2 coronavirus disease. In very recent times, it was revealed that the SARS-CoV-2 coronavirus variant B117 was able to spread rapidly across the population due to a mutation which was causing the phenylalanine amino acid residue to be converted into tyrosine residue in the spike protein sequence, which was also found to be located in the receptor binding domain. It is for these reasons that tyrosine is an interesting target for bioconjugation and also leads to a lot of information regarding the structural and functionality of different proteins. Tyrosine is generally found to be uniformly distributed in the proteins. It can either be found on the surfaces or embedded within the hydrophobic cores of protein. It can also be enriched and frequently serves as the strong interaction point in protein interfaces, which are also known as hotspots, and bind small molecules, nucleic acid, or protein partners. One very common technique of utilizing tyrosine in the study of structure and functions of different proteins is the bioconjugation or the site-specific modification. The site-specific modification of peptides and proteins by chemical bioconjugation can provide valuable information about their functions and the topology. So in our study, we have used a PTAD-based chemical bioconjugating reagent, which was used to label the tyrosines in peptides and proteins, leading to give us structural information about those particular proteins and also the functions which they are involved in. In order to label the tyrosines in peptides and proteins, the PTAD bioconjugation reagent was treated with the peptides and proteins in PBS buffer under pH 7.4 under physiological conditions, followed by the tryptic digestion, desalting, and MSMS analysis. And the results obtained from the PTAD labeling were validated using the Proteome Discoverer software. The results section of my poster discusses the multiple proteins in which the tyrosine residues were labeled using the PTAD bioconjugation reagent. Some of those proteins include BSA, carbonic anhydrase, beta-casein, and other proteins involved in the cell lysate samples. Our work explores the utility of PTAD bioconjugation reagent to tag the tyrosine residues in peptides and proteins, and we further aim to utilize the PTAD-based reagent to probe protein-protein interactions. Thank you.